Good afternoon. Welcome to the Gramene webinar series. My name is Marcela K. Tello-Ruiz. I am the project manager and outreach coordinator for Gramene. And today we're going to talk about the Biomart utility for data mining for plants. The presentation that I'm going to show you today, you can follow it in either Gramene or the Ensemble Plants website, because we share the same Ensemble infrastructure as well as the data sets that we produce in a collaborative way. A couple of tips. If your video is slow or stalling, mouse over the video window and a menu will appear at the upper left. Select medium or low quality will appear from the drop-down menu. Select medium or low quality will appear from the drop-down menu. You can also toggle the medium or low quality by clicking the icon with 4 arrows at the upper right of the video window. Click again to return to normal view. If you have any questions during the presentation, use the chat window at the bottom of your screen to post questions. There is a question. Yes, you should be hearing sound. I'm not able to type the answer, but maybe somebody will. Okay, thank you. Okay, let's get moving. So first of all, I just want to give you an overview of what Biomart is. This little tool, it allows us to mine or explore data, in this case plant data, and export the results in a simple format, tables. You don't need any programming skills for this, and this is the web interface that we use at Gramene and Ensemble Plant. However, the Biomart software, it's an open source utility, and it's also a community-driven project that provides an unified access to the diverse plant all kinds of species research, and it's applied in use in many universities and research institutions. And here I'm just showing you a small list of almost 40 resources that take advantage of this nice mining tool. It was developed in 2004, and you can read more about it in the latest publication or go to the Biomart portal. Okay, as the words, data mining, you may think of a mine, precious metals that you're going to extract, but it may also make you think about a supermarket. In a Mart, if you may want to come well prepared to explore the data available in your genomic supermarket. So you define your queries, and this way will allow you to come out with a defined set of results. So this is just an analogy to show you that this is the Biomart. Imagine your genomic supermart, and your query is going to be defined by either genomic regions, a list of gene IDs, gene types, number of transcripts, function as defined by ontology terms or interpol domains. And when you want to export your results, you also will need to be careful about defining how you want your results in a table. These may be gene features, including cross-references to other databases, gene structure, homologs, sequences, variants, genotypes, and frequency data. If you want to go directly to the utility, you can use either of these URLs to go, or if you are in the Gramene website, in the homepage, you could click in the link on the navigation bar to the left, or at the bottom under Gramene portals. You can also go click in the genome section, the icon or the URL, and this will bring you to the genome page that we share with our collaborators at the Ensemble Plants. In here, you'll see on the top, there is the Biomart link for the tool, and this is where what the homepage of the Mart is going to look like. In this case, it's already pre-filled with some information, but basically, any query is going to consist of three steps, two major steps. The first one is going to be you'll be able to choose your data set starting for a database and a species. The second one is going to allow you to use filters to narrow down the data set that you want. And the third one, you're going to specify the attributes. That is, define what exactly you want out in the exact order you want it. So gotta be careful when you click on each individual feature that you want in your result table, because that's the order that it's going to appear. And you can always trace back your steps. There are two nice features for these. One is going to be the count button that will allow you to monitor whether you are actually getting the set that you want, that you're narrowing something, and the results, which is going to give you the tables in different formats. So let me show you again in that homepage how where you'll have these steps before we go into an actual demo, little video recording for it. So first, on the top, you'll see a data set showing. In this case, I have already selected as a database the genes and as a species Oryza sativa. But I could choose also variation, and I could choose of 39 different reference genomes for genes. For variations, we have currently 11 species. The second step, if you remember, was to narrow down the set with the filters offered. And in here, you can see that this is a little highlighted in blue because it actually oh, sorry, it's showing, okay, the green is supposed to show you the filters. So the filters in this case are the region. You can specify a chromosome with the coordinates start and end, or you can go directly and copy paste a set of multiple regions that you're interested in, like we're gonna do for this example. You also have gene filters, specific, you can specify, upload a gene list, or you can specify, define the function of your genes in the results with gene ontology terms, plant ontology terms, and others. Protein domains, you can specify the interpro domains that you want your results, the genes in your results to have. And variation, you can also define whether you want variants that have certain characteristics, and I'm going to show you in a little bit the type of functionality that you can use to define your variants. The last, it was the attributes where you specify what you want in your output. And this is going to be a little more clear, I believe, when we go into the live the recording. But that's going to be a little bit fast, so I wanted to give you a heads up of what's coming. So here is an example query. I am going to use a publication, well, a set of publications from John Dobley's group. And in these, it was originally published in 1993, a QTL mapping scan for looking for plant and near architecture regions, genes, in fact. What we're gonna do today in this demo is get all the genes in selected genomic regions coming from those interesting QTLs. From those, I am going to use gene ontology terms to refine, to narrow down the functional associations with the phenotypes, the traits that I'm interested in. In this case, have to do with plant and near architecture. And then I'm going, among the candidate genes that are going to be a handful, I will identify SNP variants with a predicted stop codon gained. If we have enough time, I will show you how to also find homologs for one of those candidate genes, and we're gonna download their sequences. So here is that original publication I was talking about. This is the list, the table from the publication, or just defining the traits that they were looking at, and just to show you the kind of words that you are going to be interested. If in this example, I'm going to be interested in terms, in gene ontology terms that look for architecture, branching, and so on. So from another, a more recent publication from this group, I took out this nice table because it shows the peaks. And from the original table, I also paper, I also have the table with showing the lot scores in that very first scan. So this is only, again, for demonstration purposes. I am going to use a mix of regions, candidate genomic regions that have already been published with with a couple of these regions from these papers. Some of these I got with the kind help of Zach Lemon from John's group, formerly in John's group. So in this table, you can see some of the genomic regions, chromosome start end, that are associated, say, for this phenotype or linked, and some of the the publications that already show them. So this is kind of a proof of concept. And I also am listing here some of the gene ontology terms that I will use for the demo, because they already, and you can see that they hint already at some functionality in the architecture of the plant. Okay, so basically this is what the format that I'm gonna present, and if we have enough time, I will use the TB1, Teosinte branch 1 gene, to demo how to get to find the homologs. So again, just to remind you, first we're looking for genomic regions. Second, we are using functional associations to narrow down a subset of interesting genes that could be associated with the phenotypes of interest. Third, I'm going to identify SNP variants with a predicted gain stop codon. If we get in time, we will find homologs. Another way to look at this query is we are looking for those attributes in the middle in the intersection region of genomic genes within the genomic regions of interest with Go annotations or interpro domains that define a functionality for this region. And for this set, I am going to use the predicted variant effects from using sequence ontology terms like those in the list. In this case, we talk about stop gained. Okay, so without further ado, I'm gonna start showing this little video. If it goes too fast, let me know. I'll keep my eye in the chat, and I can pause my eye in the make it a little slower. Chat, and I can. So here we go. From the Gramene website, from the front page, you can. I show you the three ways that you can get into Mart. So in here, I went directly to the genomes page, and I click on the top for the Biomart. This is what you see first. In the data set, you can choose genes or variation, and then the species. In this case, I chose maize. For the filters, remember I have multiple regions, so I have all these possibilities, but I'm not interested in chromosomes, or it's not a single chromosome that I want, a region, but I have multiple regions, and I already have it saved in my mouse, and so I just did a copy paste. And now you can see I had 956 genes out of the total of almost 40,000 maize genes in the current assembly. So this already narrowed down my search. So in the next, I'm going to use gene ontology terms. Here, I'm going to use 2 of those that I show you, where that have to do with architecture, morphology, and also one with cytoskeleton microtubule binding. And you can see the first one gives me 36 genes when I ask for the architecture, and when I add a second gene, I do a hit again count, and this number goes up by 6. So that means 5. I'm sorry. That means there is at least 5 more genes that are associated with the second Go term, and not the first one. So okay. So anything that it's additive in this case. So all the Go terms are going to appear. Or then in the attributes, remember that I get to choose what I want in my results table. In the order that I click is that they are going to appear. If you want to change this, you can go back and click again, so you can have the order that you want. Here I have gene ID, gene name, if there is a common name that you want in your results, chromosome, start, and end coordinates. I can also select for unique results because you can see that there are duplicates. In this case, it's because the genes may have different transcripts, but it could also be that the different transcripts can have the same Go annotations and so on. So it's a nice feature to use, the unique results only. Here you saw that I selected XLS, which is the Excel format, to download my results in a table. You can also use comma separated or tab delimited tables. And so on. Now, the next thing that I would like to do is get my, well, I'm going to add, in my results, I am going to include the gene ontology terms because I narrowed down using 2, but these genes have many more. So I want all of the ones that are, the genes that I the genes that were selected have, and I'm also selecting interprodomains. So in my results table, now it's going to show up with the exact same number of 41 genes, but this time they're going to have columns for every Go term and every interprodomain that they have been annotated with. Again, if I click Go, I can get this in a table format externally, and I can browse through. And if you see in the gene name column, I can you'll be able to see the Teosinte branch 1 gene, for example, that it's in that was already published for these QTLs. Now I'm going to use a second data set, and the choices are, for the same species, I could choose variation, which is what I'm doing now, but I could also choose instead a different species and for genes. So, but here, what I did before, it was maybe a little too fast for you, but I copy-pasted the genes, the list of 41 genes from my one of my tables, and I use that here. I copy pasted on the gene in the gene list. Those are genes table IDs, and I'm using that. I'm also asking for a stop gain in any of those genes in the list. Okay. So here my results. This is what I want in my table. ID, the source, population if there is, and anything that is interesting to you, you can choose at this point. And again, you could add to make sure that they have a stop gain. You can select the consequence to the transcript, and let's see that we get what we wanted. Yep, we have a set of 9 variations now. At the end, we ended up with 9 SNPs and these 9 genes. And so that that is what you can do. We're almost at the end. I think I'm gonna leave it here. I'm gonna go to the next variation, so we have time. I'm gonna show you here also in the beginning. We are gonna start with the tyrosine branch 1 gene, TB1, to find homologs in other species. I'm gonna use closest species like sorghum, then Oryza, and a little farther Arabidopsis at the end. And when I get to Arabidopsis, I'm going to select the homologs that I get for the TB1 gene, and for those genes, I will get their sequence. Okay, so let's get started. Okay, so I already selected the genes in this frame, and their filters, I'm gonna put TB1, and I see that there is 0 genes. How's that? Oh, because that's not a stable ID, but it's actually a common gene name. So I change the setting here, and I specify, and now it gives me, it tells me that yes, there is 1 gene that is called TB1. And I am gonna select homologs in my attributes panel. And here I have, I choose a stable ID, again the gene name, because I like to make sure that that's the gene, my gene of interest. And then I go to the species I mentioned, sorghum, and I want to see what kind of homology they have, whether it's 1-to-1, there is 1-to-many homologs in the other species, and the percentage of identity, protein identity that there is between my query and my target. So I selected those 3, and as you can see, everything that you're selecting, it's appearing on the left navigation bar. So here are my results. You can see there is a single gene, just repeated 3 times for sorghum, for Oryza, and 3 different ones for Arabidopsis. This is just the way so they can fit nicely in the same table. You can see the percentage of identity is a lot higher in sorghum than followed by rice or Arabidopsis, not too much, from between 19 and 21%. So, but I am going to select, I'm going to copy paste those 3 Arabidopsis genes, and the last thing I'm going to show you in this little demo is how to get sequences for them. So I'm going to go down here as a second data set. I'm going to say I want this Arabidopsis. You don't have to do it in the same, in the second data set. You can do it in the first one. Get a new fresh page and just do that. So the important thing is under attributes, you're going to select sequences, and in filters, I'm going to put the 3 gene IDs that I have. Those are stable IDs, and as I said, attributes, I need to select sequences to be able to download the kind of sequence I want. I can choose splice cDNAs, exons, 5' UTRs, anything, gene, transcript. And I can also specify whether I want a certain number of nucleotides upstream, flanking upstream or downstream of the gene. In this case, here is for those 3 genes. And I'm gonna do a little test. I select the first few bases. You can see they are at the very beginning of that cDNA. And I'm gonna copy, and I'm gonna find that to corroborate to that when I request about 200 upstream flanking sequence, I get that same sequence, but not at the beginning, but downstream, and there it is. So from here to the beginning, there should be 200 nucleotides upstream of this cDNA. And basically that's that. I'm gonna, because of time, I'm going to go to the next slide. Just to recapitulate what we did, this is kind of the stepwise demo for any Biomart that you use. you're gonna first go to the page, to the home page. Well, you will get to choose a database from the drop-down menu. Select the species that you want to query, and then you're gonna have a filter section where with different panels. You need to click on that to display the options, and then this is gonna expand all of the options. So don't get disappointed because at first you don't see everything is clickable. And again, the results, I forgot to mention that many of those, all of the links, all the things that you see highlighted, like genes or SNPs, you can click and go to their to the page, the respective page. Okay, so then you can number 6, select filters or paste a gene ID list. You always remember to count to confirm that the filters that you choose are making sense. And the attribute is going to allow you to select what kind of features, like features or structure, or homolog, sequence variation. Those are the options that you have to download in your results. Then you can expand individual panels to select what you want to include, and when you click on the results, you're gonna be able to select how to view it, if you want to export it, the format that you want it, and just remember to use the unique flag so you can get remove duplicates. Here is a little example that you can try at home. You're gonna have, as I said, these slides available, so you can try to replicate this example by narrowing down, looking to find transcription factors having stop-gained alleles. We already use the stop-gained. You can change that to stop-lost alleles. You can use the interpros you want, or you can use Go terms instead, because there is things for transcription factor, DNA nucleotide binding, and that sort of thing. For help, additional help, you can go to the Gramene Outreach websites where we have links for the video tutorials like this, and the Gramene YouTube channel. We also provide the slides and documentation in our FTP, and the links are in that same page. For Biomart specifically, I already mentioned there is a project on its own. You go to the biomart.org and you'll get to see all kinds of resources and species that are using this powerful tool to mine data. Ensembles also have very nice tutorials, and you can get another sense or further examples to work on your own. I apologize. It was Dr. Ganesh. No, I can't remember. Sent me an example, and I did not have. I will not have time to go into it, but I will be happy to correspond directly with you, and we can solve the exercise. Last, you can follow us. We can send an email for feedback. We will certainly respond. We have a Facebook page and a Twitter handle as well. Here is the people that work on the project, and in the little squares, we have the groups that do actual work with Biomart and genome data. Thank you. If you have any questions, please use the chat box, and I'll be happy to respond. We have someone typing a question or comment. Anything is valid. When you are looking at the database for SNPs, what is the data source? Varieties of C. It's CMASE, the reference assembly version 3 currently, so it's only one variety. Maybe you're talking about variation. For variation, there is a number of variants, and they are specified. We currently have 2 data sets. One is the HAP map, the maize HAP map, and that one has, I believe, it's 104 lines, different varieties, including a handful of teosinte lines. And then there is the Pansia 2.7 GVS, genotype by sequencing. That's also available from the pansia.org website. And that includes close to 17,000 lines. You can, if you go to the Gramene genomes page, select the maize, and there is a section that describes more details about, specifically about variation in maize. That will have even references and URLs. So Dr. Ganesh Thapa, yeah, what about homologs? Homologs, we only have for wheat, we don't have it enabled in the Mart. You can find those with the genome browser, and I can go with you. I can go in more detail with you over emails. We can have a screenshot, and we can certainly plan for a future webinar focusing on showing the. This has been touched in a couple of past webinars that have reviewed the genome browsers. But if you need a little more detail, we can go back. You can explore some of the past genome browser webinars from the Gramene YouTube channel, and that will give you at least a hint of a flavor of how to get those. There'll be at least a few slides, 2 or 3 slides, but we can discuss in more detail later. Can we share SNPs with you at Gramene? Yes, absolutely. We actually have an ongoing collaboration with the European Variation Archive. It is quite important for us to try to get sort of a plant DB SNP, because we are currently not lacking. Together with the Ensemble Genomes Transplant and the EVA, European Variation Archive, we're trying to recruit as many SNP data sets that are, that become publicly available, so we can have a central repository. EVA, will be like the DB SNP for plants. That's our idea. And Gramene and Ensemble will be able to put these in tools such as Biomart and the browsers, so you can get pretty pictures and explore the data and do downstream analysis. Also, iPlant is part of this collaboration. So yes, please, if you have any SNP sets that you would like to share, that is, it's about to go public. You submitted your manuscript, and you want to include a URL with either pretty images or simply a repository, feel free to drop us an email. The requirements are pretty simple, just a PDF format in an unknown assembly, and metadata for your samples. Excellent. Thus, Gramene is encouraging the communities to submit their SNPs data into EVA, and Gramene would pick them from the EVA repository correctly. Yes. you have phenotype data. Currently, we only have expression data coming from the Atlas Express from again our collaborators at the European Bioinformatics Institute, EBA. And yes, you can find those through the gene pages, the browser, as well as the plant reactome, if I'm not mistaken. But it's only expression level at this point. We have some in our archives. We do have some trait data, QTL markers, and that sort of thing, but that's not maintained. It's not up to date. So it is provided as it was originally submitted to the database, and it hasn't been brought to current assemblies. So just go to archive.gramene.org. I mean, those associated to variation data genotypes. No, I don't believe so. We do have the capability in the variation pages, but we haven't put those. They are not ready. They are not out in the public. But it is in our plans. Yes, associations like phenotype, genotype associations to provide those to the public. Can we see linkage of gene with QTL? We know, as I said, just the only QTL data that we're managing is that we have in store is in archival form, so it's only legacy data.